Thank you so much, Ben, for the very kind introduction, and for Vitaly and Ben and the team for inviting me to share some of this work today. I just want to start by saying that this work, there's a lot of people behind this work that have supported us, collaborated with us over the years, and none of this would have been possible without them. So even though I'm presenting it, it's really a work that's done by many. So Ben mentioned that I'm titling the talk, New Developments in TB Genomics and Data Integration. So just first, just to disclose that I'm a consultant to the Foundation for Innovative New Diagnostics, and also funded by NIH, the National Institute for Allergy and Infectious Diseases, for the research that I'm conducting. And none of these represent conflicts of interest that are relevant to this talk. So just to start and say that the pace of TB genomics is really astounding. So looking at NCBI earlier today, just between 2000 and 2010, when really sequencing became a thing and became possible to do at a massively parallel scale, only about, short of 400 genomes were sequenced. But over the last decade, we've seen over 108,000 TB genomes that are now in the public space. There's a lot more data that's not necessarily public. So just to give you a scale of really how much sequencing has become a workhorse, and it has so many different application areas. And the use of sequencing, of course, is not limited to TB. It's actually become part of how we do surveillance and how we study pathogens, across the board. So example is, SARS-CoV-2, there's already over 3 million, close to 4 million SARS-CoV genomes on GISAID available today. And this is, again, due in part to the perceived usefulness, which I don't think is just perceived, I think they are useful, of, sequence data, but also because of access to sequencing tools has become more widely available. The cost of sequencing has become much cheaper, and also the know-how and the ability to process sequencing data is definitely improved and increased over time. So ultimately, what many of you know this, but just to kind of level the ground for folks who are less familiar with sequencing, really the benefits of sequencing are that allow you to measure differences and compare genetic sequences. So in this little toy character example here, strains 1 and strain 2 are more similar in the sense that they have a similar base at this position highlighted in red, and strain 3 has a mutation there relative to the other two. And so you can reconstruct that those differences using kind of a phylogeny or other kind of comparator or distance measure. And this allows us to really understand and recapitulate the relationship between strains, and that's useful. No problem. I was just talking about how, pathogen strains evolve over time, and the mutations that they accumulate help us kind of reconstruct transmission networks, or help us understand changes in the organism over time. And what's underlying the right. That's never happened before. That's quite interesting. Okay, so what I was trying to say here in this plot is that the reason why these mutations are actually so interesting and useful is that they, in general, a kind of a rule in biology under neutral evolution, is mutations tend to more or less accumulate at a steady pace over time. Of course, this is sort of a generalization, and it's more of a useful approximation than it is necessarily applicable to every gene and to every strain at every time. But just to demonstrate this point, I'm plotting using nextstrain.org SARS-CoV-2 mutations over time, plotted from the beginning of the pandemic up till I believe this was in early 2021. And you can see here that if you plot the distance from the root, and you kind of here are coloring all the different strains by when they were isolated, you can see that clearly the strains that were isolated earlier were more close to the root, strains that were isolated later were further away, and the rate at which variation was accumulating was roughly 25 substitutions per year. And so this property is very useful to try to reconstruct evolutionary histories and reconstruct transmission networks. But beyond neutral evolution, organisms evolve because of different pressures on them. So to give another example here, under antibiotic resistance pressure, when you have mutant isolates that have the ability to resist killing by the antibiotic, they are selected for, and then those actually become more prevalent and essentially form the next generation. And these forces often represent themselves in forces of positive selection that we can look for in genetic sequence data. So that essentially the innovation here is that you have the ability to take clinical isolates and then kind of reconstruct evolutionary history and study the biology of the organism. So previously we were restricted when we studied organisms to doing a lot of in vitro based work, but whole genome sequencing really made it possible to study organisms within their host work, where we use, so ultimately the goal is really to use this genomics to help us better diagnose disease in individual patients, but also help survey disease across populations. And in our previous work, we've been quite interested in this idea clone of a particular clone of lineage 4, and that actually predominates in Peru in our data. It's also been described in Spain and Portugal upon our review, and it is actually a sublineage. So this phenomenon of seeing these frame shifts isn't actually convergent. You don't see it happening repeatedly. It only happens in this particular lineage that happens to be pretty common in Peru and actually happens to have a very high rate of MDR. So this phenomenon is of value, because if you try to then use MMPR mutations to predict resistance, you might deprive patients of an important drug if you interpret the results inaccurately. And so we found that pretty much the majority of these isolates carried both of these mutations. So those results are now published in AAC. Okay, and so really the goal is to take information that we've gathered around, what mutations cause what phenotypes, and be able to integrate them into a model that allows us to kind of, in an automated way, interpret the genotype and really be able to produce as accurate predictions as we can. And as many of you know, for TB, this is very important because it takes forever to grow the organism. And it really takes even more to actually do drug susceptibility testing. And in many parts of the world, that's not even possible because of limited capacity and cost. And so this really, right now, I think a lot of people are hoping that whole genome sequencing will continue to be the answer. And so I think work and investing in doing work in this space is hopefully going to pay back dividends as far as our ability to use this information to diagnose and better treat patients. So we've also been interested in this space of, okay, we like to study mechanisms and do genome-wide association of specific resistance phenotypes, but we're also interested in then developing some analytical framework that would take the genotype kind of put it in some kind of black box, spit out a resistance antibiogram or resistance profile. And so in this study that was funded by the Gates Foundation, and Megan Murray was the PI of this study, multiple national, supranational labs contributed that were sequenced using a targeted sequencing approach called molecular inversion probes, which targeted specific 18 different genetic and their promoter regions. And so this just kind of depicts the origin of these isolates in different parts of the world. We didn't have whole genomes, so we couldn't actually call specific lineages at the time. So we called this now archaic concept of principal genetic groups. And but we did construct a phylogeny using the drug resistance data alone, and it did actually recapitulate what we expect that structure to look like, although probably not perfectly compared to a whole genome tree. And here I'm just demonstrating that the majority of these isolates were lineage 4, all kind of represented in varied shades of red, or and then the blue isolates are lineage 2, and the purple are lineage 1 isolates. And we had a very high proportion of MDR strains in this. And the ones that are labeled with these small s's represent susceptibles. So there was approximately 1,200 TB isolates in this study, and about 900 of them were MDR. And then we really wanted to learn, because this was, among the largest data set sequenced to date of TB isolates. We really wanted to learn from the genetic sequence data of what genes mattered, what mutations in the genes that we sequenced matter, rather than kind of go at it with a certain list and see whether or not that works or not work. So to do that, we actually used a random forest approach, which is now called machine learning. It wasn't called machine learning at the time we applied it. But essentially, it's an approach that allows you to study individual mutation, or in this case, features, and kind of rank them by importance. And it's actually kind of widely accepted as one of the better tools to pare down or kind of rank features by importance. And that's why we applied it in this case. And it actually operates as essentially constructing these trees that decide on what mutations to put in each of these boxes. And there's a lot of dependencies in these trees. So, what question comes after? So what mutation comes after on the next tier depends on what the performance of the mutation as a classifier on the first tier, and so forth. And so in that way can actually incorporate interactions between mutations to some extent and its effect on phenotype. And so these models actually construct multiple trees, then they take a vote across multiple trees, and that's why it's called a forest. And the randomness comes from the fact that features are sub-selected at each step, and the data itself is bootstrapped or sub-selected to kind of stabilize the model. And so we used random forest to actually and perform this feature selection. And just to show you here that what we did was we initially constructed one random forest on all of the mutations that we had in the INH resistance genes based on essentially a Delphi panel where those genes were decided upon before they were even sequenced. And so this was the original model over here that had the most number of mutations. But then we sequentially pared down the model, removing the least important mutation at each step, and kind of measured sensitivity and specificity. So specificity is in red, sensitivity is in black. And you can see for INH that paring down the model really doesn't do anything in terms of the performance is still very good. But really once you get down to kind of close to 20 mutations, so that's what we showed, at least was able to achieve the best model performance on the data set that we had, which was very heavily skewed towards MDR. We had much fewer mono INH resistant isolates in this data set, relatively speaking. And for RIF, it was also a similar story. The specificity was high and the sensitivity was high, and we really didn't need that many mutations to make a good prediction. For pyrazinamide, pyrazinamode's always interesting. you actually couldn't par down the model very much. You would quickly start to lose accuracy. And we noticed, even though you can't quite see it, that there was a lot of fluctuations in performance as you par down the model. And this is because individual PZA-resistant mutations are pretty rare. And so because of the bootstrapping that happens and the subselection, you sometimes see an effect of dropping it and sometimes you don't. And so essentially for PZA, you need over 100 mutations to do a good prediction, but that prediction wasn't that good. I think we achieved just a little over 60% at the time. Now our models are better. So just to kind of summarize that particular result, so we were able to, and as many of you know, we can do predictions for first-line drugs with reasonable accuracy, but our accuracy drops down for second-line drugs. And at the time, we showed that you actually need hundreds of mutations to actually predict resistance. We were trying to make the case that you can't really do targeted sequencing only, and that you would ideally do whole genome sequencing. I think that's, there's still multiple targeted platforms that are being developed for direct from sputum kind of resistance diagnostics, but they will have to have a high multiplex of ability to capture many mutations for them to be accurate, or potentially, a lot of diagnostic companies have, for example, given up on pyrazinamide entirely because of this phenomenon of how many mutations it requires to predict resistance. But since then we've been doing more work here in this space using different types of statistical model architectures and more classic kind of machine learning type of approaches to predict resistance. So in one study we applied a neural net, but kind of modified it to construct something called a wide and deep neural net, where we acknowledged the fact that sometimes you need to capture interactions, for example, for drugs like ethambutol, maybe pyrazinamide. But for other drugs like isoniazid and RIF, we do pretty well, like, even with simple lists of mutations. So maybe we don't need that complex architecture. Maybe that will introduce unnecessary noise or overfitting. And so this allowed us, we took this approach where you can kind of bypass the neural net, and that's essentially, it's like a simple regression, and or you can kind of go through these 2 layers of nodes that allow you to capture epistasis or interactions for other drugs. And we showed here that there were some modest benefits over other models, certainly over regression and over random forest. But the key ingredient wasn't even the model itself and how it's built, whether it's a neural net or a random forest. That mattered less than actually what you put into the model. And so this is sort of what this slide is trying to show here, is that you do, for example, observe some gains for, just to point here to fluoroquinolone, say, to turn your attention to MOXIE. So if you only put in known resistance mutations, you get kind of a lowish accuracy. But if you put in mutations that actually encode resistance to other drugs, the model can actually predict fluoroquinolone resistance more accurately because it's learning dependencies between the drugs. And that actually can be useful, although it's yet to be proven how generalizable that's going to be. So we're still kind of worried about that piece. But also for certain drugs like pyrazinamide, you really should be including not only common mutations in the known causative loci and other loci causing resistance to other drugs, but also you have to make sure that you include really rare variants. And that actually results in the largest boost of performance for drugs like pyrazinamide. And so we have actually over the years built this online tool that allows users to go in there, you can deposit a FASTQ file. You wouldn't deposit it, you essentially run it through the pipeline. You can pick whether or not you want to apply the random forest model or this WDNN model that we've built, the wide and deep neural net, and you can get predictions. And if you want more details on the accuracy, we've recently validated this tool against microbe and TB profiler, and our tool has higher sensitivity, although at the cost of a very small decrease in specificity compared to microbe and TB profile. So So if you have an isolate or a patient who's, doesn't seem to be behaving like you expect them to, you should try to give this tool a whirl and see if it actually predicts something more than you've seen with other tools or with your internal workflows. And certainly would love to hear your feedback on it. Okay, so, along the way, as we have, well, maybe I could stop here and for a second and ask Ben, I know we kind of lost some time, so should I try to kind of go through quickly some parts of my talk and get to the end, or
I think you can
extend beyond 10?
You can continue as you planned. It's really important and interesting, and we've got our audience. It's better for people to really hear the full story. Hopefully we'll get.
And you're recording it, right? So yes. I guess if you have to leave. Okay, sounds good. Okay, so along the way, as working with more and more sequence data, we've become quite type A about accuracy using whole genome sequence data, and we've kind of developed our own pipeline. I'm sure many labs have developed their own pipelines, but we've been interested in benchmarking tools and kind of learning what is the best tool and how much of the genome can we actually study. And as some of you know, for example, classically, when we remove something like 10% of the TB genome because it has these repetitive regions and the PE-PPE genes, and for example, the CDC does this routinely, and then the portion of the genome that's you're left over tends to have much less diversity. And as far as like using that information to kind of maybe trace transmission or assess outbreaks, you have much lower resolution. So ideally, we would want to be able to retrieve as much of the diversity in the TB genome as possible, but do it in an accurate way. So in this study led by Max Marin, a PhD student in my group, we actually collaborated with the National Jewish Health, where we sequenced isolates both using Illumina and PacBio long-read technology. And we hoped that we'd be able to complete the genomes and be able to then study the accuracy of Illumina and benchmark it against complete assemblies in TB in these clinical isolates. So we had in the end we were able to circularize, essentially completely finish 36 TB genomes. This is their phylogeny. Again, as is commonly the case, lineage 4 predominates, but we do have representatives from lineage 1, lineage 2, 3, 5, 6, and even 8. We actually downloaded this from a study that was recently published on lineage 8 and included it. And so our assemblies show essentially good accuracy. We find very, very high average nucleotide identity to H37RV. We find that in general, TB genes are pretty monomorphic. None of this, potential concern that the TB has this large accessory genome that's emerged intermittently in the literature. It is, as we expect it to be, quite clonal and quite conserved. And so we used these completed these complete circular assemblies to benchmark accuracy of Illumina. And we did that by actually looking coming up with this metric that we call empirical base pair recall. And essentially this measure essentially is, for you to have, for a site to have high empirical base pair recall, it has to be both confidently and accurately called, meaning that the whole pipeline had confidence in the call, and the call, when compared with the ground truth PacBio based assembly, was correct. And so you can see here, here we're plotting this EBR measure that I just described, as well as mappability, which actually gets at the repetitiveness of the sequence. And then in yellow here, we have the GC content. And so you could see that there are certainly GRS genes that are quite problematic with really very low empirical base pair recall. Usually that coincides with repetitiveness and high GC content. And so, but you could see that there are obviously the majority of TB is not like that. And so we studied actually these regions that are classically removed. We kind of used the Coscola et al. Definition of them that was that was a well-cited paper around what, how these genes are defined. And you can see here that compared to genes that are not traditionally excluded, so or high confidence regions, certainly the putative low confidence regions, which are essentially, we break down into repetitive regions, PEPPE genes, and insertion sequences and phages, do have lower empirical base pair recall and lower mappability scores. We then kind of compared the accuracy of SNP calling when you just exclude these regions, and compare that to a schema where you exclude just low mappability regions, not, which is sort of a less strict masking or exclusion. And then we compared that to our pipeline where you don't do any exclusion. You actually just rely on the variant caller accuracy measures, and specifically the mapping quality MQ here. And so you could see here that just kind of at a very high level, you could see that when you exclude these regions, you have very low recall. Like you're kind of missing something like 30% of the variation in TB, which is a substantial amount. Even the best schema couldn't achieve more than 89% using Illumina, which I think is sort of a lesson that I shouldn't call best, right? The most permissive schema couldn't capture more than 89% of the variation in TB. And this is because we know this, like TB is quite repetitive and Illumina can only do so much. But I think what we demonstrate here is that excluding these genes is very, very conservative. It's too conservative. In fact, if you decide to go with the pipeline that we use, which we've actually now compared to other pipelines, even though it's not currently in the preprint, we've actually done that in the revision that the peer reviewers requested. This pipeline that we use is actually superior to other pipelines when compared head to head. But if you do use that and you use the mapping quality, you have you only lose a very little precision. Again, this scale is quite blown up. You lose something like 0.4% precision, but at the expense of gaining like 15% in recall. So it's definitely worth it. Obviously, depends on the application that you're using. But if we have actually proposed a new masking schema for those who don't actually want to trust the variant caller and still feel that they want to preserve precision at all costs. And so if you go to our GitHub page, you can find essentially this page, MTB Illumina whole genome sequence evaluation, that actually gives you a bed file that you can use to exclude regions of the genome where we see the false positives and where we see low mappability. That gives you a slightly lower F1 score, mostly because you lose some recall, but you have very high precision. Although I should say that this precision was obtained on the same data, and potentially if we obtain independent data, it probably will be slightly lower than this incredibly perfect number. But again, all the details are in the preprint. Okay, so I'm going to proceed and talk about genomics for surveillance. And so we've been quite interested in, as Ben mentioned, we like to bring data together and integrate data. And we've used that to build some of these resistance prediction models that I described to you before. But we've also used it to kind of understand TB transmission globally, and to also kind of come up with new prevalence estimates. And so in this study, we curated data from the public space, and we wanted to ask this question of how and when did drug resistant TB develop in different parts of the world, and compare countries as far as when they acquired resistance to different drugs, and actually learned a few things along the way. And this study is a kind of a little bit of a forensic public health, because we're kind of going back really far in time. and kind of reevaluating, TB drug resistance transmission kind of over the last few decades since antibiotics came in. So this, the basic idea of how we did this is that we used molecular dating that relies on this concept of a molecular clock, and that it ticks at a specific rate. And for TB, that's a well-known rate that's been kind of reproduced by multiple investigators over time. And we used this molecular clock date as well as the phylogenies that we constructed for each country and lineage and for each type of drug. So for isolates that were phenotyped for a specific drug, and we're able to date when resistance was acquired to each drug in that country, in that lineage. And this was sort of a computationally massive endeavor because it involved, processing over 10,000 isolates and then deciding when we can or cannot date for every particular country, because we had strict criteria, because we required the sample to be balanced between resistant and susceptible isolates in each country. I won't go into that into a lot of detail, but if any of you have any questions, definitely let me know. And this paper is published in Lancet Microbe earlier this year. So, but to summarize, like kind of the main findings that we found that resistance, when you look at across all countries, across all lineages, on average, INH resistance was acquired first, followed by rifampin. And I'm showing you here the overall order. So this is just showing you that when you compare the dates at which INH resistance was acquired compared to RIF, it's statistically different. So this is what the red here represents, and these are the p-values. And so we could see that actually INH is older than RIF. RIF comes after, and then just to kind of show, comes after, but actually comes around the same time as streptomycin, which was an odd observation, and then ethambutol and PZA, and then other second-line drugs. And this was quite interesting why we observed streptomycin to be acquired so early, because we were expecting to see streptomycin being acquired first, because it's the first antibiotic to have been used for TB. But really, in the end, the best explanation for why we observed streptomycin resistance acquired at the same time as RIF is because of essentially this practice that was prevalent in the 90s and early 2000s, and is actually still, I think, hopefully has become obsolete because it's no longer recommended by the WHO, but it's this practice of adding streptomycin to a failing regimen with patients who come in who have failed drug-susceptible TB treatment, who then kind of get represcribed drug-susceptible TB plus streptomycin. That's the most parsimonious explanation of this observation. But I think this kind of demonstrates how you can do a little bit of forensic public health and realize that that was clearly bad practice, because that pretty much just breeded strep resistant without necessarily helping patients get cured from their disease. And one other more recent surveillance effort that we've embarked on with support from the World Health Organization is we conducted essentially whole genome-based antibiogram prediction by country for TB. And so, as many of you know, TB drug resistance estimates or true prevalence estimates are really hard to come by, because few countries actually conduct regular drug resistance surveillance, especially, focusing here on high burden countries. Obviously, the low burden countries tend to conduct surveillance much more regularly, or it's really part of what they do for patient care. Increasingly, the advent of the nucleic acid amplification tests, including the export, has made that more easy. But and there's more data kind of being generated to kind of monitor RIF resistance, but it doesn't monitor resistance to other drugs more regularly. So we reasoned that if you use, what if we take everything that's in the public space for TB genetic data and see if we can do some bias corrections and see if we can come up with a full TB antibiogram for each country. And this was work that was done by Avika Dixit in my group. And so, so essentially the answer was, so these are several kind of different considerations that we made while doing this work. First, if you take the whole genomes, it's rare that, it's not rare, but it's not common that you always have phenotypes. So we had to actually use some of the models that we've developed previously to predict resistance directly from genotype. And that in some ways, circumvented this idea that we don't have phenotype, or the phenotype may not be standardized, et cetera. So we used genotype-based prediction across the board on the data that we were able to gather. And then we reasoned, because of this idea that TB sequencing is often used for outbreak or transmission inference, what if, the data is really skewed towards outbreaks of resistance? There's certainly been several studies published in the kind of Eastern Europe, former USSR region, that have really shown how clonal drug resistance is in that part of the world. And they've kind of focused on XDR, for example, studies that have only focused on XDR in South Africa. So how do you circumvent this oversampling? So we actually developed a method whereby we exclude essentially isolates that are really close genetically and focus on more independent observations that are much less likely to be related due to recent transmission. And also the mere fact that, because, for example, in our previous work, and I'm sure many others, we sequence TB when it's resistant because we want to learn about resistance. And so that's also a consideration, an important one, about oversampling of resistance in the public space, and that we need to be able to correct for that. So the way we did that was we actually conditioned by the WHO reported RIF resistance rate. We argued that that's more accurate because it's coming more from expert-based surveillance that's become more prevalent. And so everything was corrected to the WHO RIF resistance rate. And then we also corrected for the fact that we're using this machine learning model to predict resistance. And so we corrected for the fact that that model has imperfect sensitivity and specificity. So just to show you the data that we finally ended up with. So we kind of initially gathered 24,000 TB genetic genomes that had geography. So we had country of origin data. We excluded 1,000 of them because their genomes had a lot of missing sites along the length of the genome. We then exclude the further 3,700 that were really close genetically. In the end, that exclusion didn't make a big difference on our resistance estimates. Just want to put that out there, but we did do this exclusion. And we ended up with a total of 20,245 whole genomes that were gathered in over 20 countries. And we also had phenotype for some of them for about 12,000. We also studied that in the manuscript that we're about to release, actually. So we also took the predictions that we made and compared them to national survey data. We did that specifically for South Africa because there was a lot of whole genomes from South Africa, and we actually had access to the details of their national survey that was phenotype based. And we found that our estimates, which are in gray here, kind of compare very favorably to the estimates from the National Drug Resistance Survey. And you should ignore RIF because we actually corrected for that based on what I mentioned before. But for the other drugs, they were within the confidence interval, with the exception of isoniazid, where we were underestimating isoniazid resistance. And that we think is related to the fact that most of our resistance models have been built on MDR INH resistance, not necessarily INH monoresistance, which is probably much more diverse genetically. So that's sort of an Achilles heel here. But this is, at least we learned something about that these models are not yet up to snuff for INH monoresistance, which is the most common form of INH resistance in South Africa. It isn't the case everywhere in the world. And also for aminoglycosides, which are now becoming obsolete, but we were underestimating resistance slightly to these drugs, not overestimating. Okay, so one of the major findings of the study that is that we found actually that fluoroquinolone monoresistance is actually substantial in the Southeast Asia region. And considering, the imminent rollout of the fluoroquinolone-based regimen for drug-susceptible TB, the 4-month regimen, we think this is a notable finding. And I think it's going to be really important to be surveying for more resistance amplification if this happens, if these drug regimens are rolled out in Southeast Asia. Again, the percentages are not very high, but the mere fact that they exist at this level among these are RIF susceptible isolates, it's really concerning because you wouldn't necessarily be capturing these by doing, just an expert test. Okay, and you are welcome to explore these ANTIBIOGRAMS that are now on the web. There's probably some cosmetic things that we're still working on, but the data is all there and accurate. And so this is also on GenTB. We kind of navigate the maps and then to ANTIBIOGRAM, and you can kind of look at these estimates. We computed 3 types of estimates: marginal rates that are essentially across all TB, and then rates among RIF susceptible, rates among RIF resistant. And with that, I just want to thank my group, the lovely group in the, this is a picture of them in the COVID era, celebrating a colleague, and also our funders and our collaborators and my department leadership, which has been incredibly supportive. And thank you all for listening. Happy to take questions.
Thank you, Maha. That was really an amazing overview of the progress that you've made. And clearly, this is providing a whole new level of sophisticated insight into the global epidemic. I'm wondering, it may not be possible for us to take too many questions, but if you don't have a problem, if people were to contact you directly, if they had specific issues to clarify with you, is that?
Yeah, absolutely no problem. I realized I did not put my email on the slide. Apologies about that, but it's not too hard to find me. I'm also on Twitter. You can feel free to me directly.
All right, so maybe we should do that instead of me picking 1 or 2 questions. I think it's probably appropriate, given that people will have other appointments at 10, to wrap it up here, but to just thank you that despite this disruption, you were able to really provide us with a very insightful overview of where things are at. And hopefully we can follow up on this and keep track of the work from your group in the future and become partners in some of your new endeavors. So let me just—
Thank you so much, Ben, for the invitation, for all of you for listening. And yes, please feel free to reach out with any questions and discussions and feedback. I love to get it, and this is why I do what I do. So thank you all.
So thank you. That's been very inspiring. I think Vitali had to leave us to go to another meeting, but I should just thank Vitali and Chris from the TB-CRE for having helped to organize this, and for all of you from around the country that have joined us. I think this will really help to guide how we steer and set things up in Australia as well. So we'll definitely be in contact. Thank you once again, everyone, for joining us this morning and for staying up late at night on your side.